You want to go ahead and start?
Sure.
Or over.
All right. Good afternoon, everyone. Welcome to Endocrine Grand Rounds. It's a pleasure to introduce Dr. Lisal Folsom. Dr. Folsom graduated from Loma Linda to get her medical degree. She then went on to do a combined internal medicine and pediatrics residency program at the University of South Carolina in Charleston. I'm just getting the nod to make sure I'm doing that correctly. And then she then went and got her fellowship in a combined fellowship also in pediatric endocrinology and adult endocrinology at Indiana University and Riley Children's Hospital. At the same time, she also obtained her Master's of Science in clinical research. And so she joined our faculty, joined us here in Louisville in 2017, where she has practiced both pediatric and adult endocrinology. Her clinical specialties, she has focused a great deal on transition in care. And so this has been a big area of her practice, as well as gender care in our pediatric endocrinology office and in the adult practices. And she has also been a tremendous educator, both for our patients, families, and our providers on healthy lifestyle, including healthy eating. And so she has certainly taught me a lot about the fact that I should continue to work towards more of a plant-based diet. And so aside from my turkey sandwiches, I'm working towards that pretty well. And so I think this topic is perfect for her for sharing today, and it's cake or death, the erythritol controversy. So please welcome Dr. Folsom.
Thank you, Dr. Wintergers, for that very kind introduction. I am so excited to talk to everyone here in person and online about the erythritol controversy. And I have entitled this talk Cake or Death for two reasons, one of which will be revealed at the end of the talk. And the second of which is that, do we have to choose? Do we have to choose between healthy nutrition and enjoying the food that we eat? And hopefully, during our time together, we'll be able to answer that question. The reason that I chose this specific topic for my grand rounds is that several months ago in the journal Nature, a study was published looking at erythritol and potential cardiovascular risks. And this study was picked up by the media, and there was a lot of consternation and a lot of worry around the both medical and then regular community about erythritol and whether or not this contributes to cardiovascular disease. And so in the context of that article, I wanted to really do a deep dive and see whether this is something we need to worry about. So I have no financial disclosures. However, I do have some non-financial disclosures to make, and that is things located in my house. So these are some photographs that I have taken of some products that are located in my pantry, and you can see that all three of these contain erythritol. So this is a topic that is personally relevant to me and my family. During our time together, we are first going to talk about the history of erythritol and its current role as a non-nutritive sweetener in human food products. We are then going to talk a little bit about some biochemical properties of erythritol that may or may not be contributing to potential cardiovascular effects. Then we're going to look at the health data that existed prior to the study published in February. What is erythritol based on? Why do we use it? How do we know that it's safe currently in food products? And then we are going to spend quite a bit of time talking about the study that was published in Nature. And then finally, we'll be able to develop strategies for how to deal with erythritol. What do we tell our patients? What do we tell ourselves? How do we move forward with this new information and any new information that comes out related to something that we thought we already knew about? So erythritol, It was developed or discovered in 1848 by a Scottish chemist named John Stenhouse. And this chemist really enjoyed looking at plants and finding new uses for the chemicals that he discovered in them, particularly focusing on medical and technological developments for substances that he could identify in the plant world. During his time in South Africa studying a lichen called Rossella montagni, he identified these clear crystals. And when he tasted them, as chemists sometimes do, he determined that they were very sweet. And this was erythritol. Not a lot was done with erythritol as a substance until about 100 years later, in 1950, when they were looking at blackstrap molasses that had been fermented by yeast, and they identified this same substance that John Stenhouse had discovered 100 years ago. And since the 1990s, erythritol has been used by the food and drug industry to sweeten foods and beverages in various parts of the world. So erythritol has some unique features that cause it to be useful as a non-nutritive sweetener. And the biggest is that it doesn't have an aftertaste. A lot of people will say, you know, I can't use NutraSweet, or I can't use Splenda, or I can't use Sweet and Low because it doesn't taste good or it has an aftertaste. Erythritol does not have this aftertaste. The other unique feature is that because it's crystallized in form, it can provide volume and texture to baked goods, similarly to white sugar or granulated sugar. In 1999, a joint committee of the FAO and the WHO on food additives determined that there was no acceptable daily intake for erythritol. But in 2003, the European Union Scientific Committee on Food did conclude, based on some data that we'll look at, that erythritol was safe for use in foods. And as we do, the US followed several years later, and in 2018 determined that erythritol fell into the category of generally recognized as safe for food products. And since 2018 in the US, it's been added to foods. So the biochemistry of erythritol, this is something that we need to look at because it may contribute to some of that cardiovascular data that we're going to talk about. Erythritol is a chemical structure known as a polyol. It has 4 carbons, and it's soluble in both water and alcohol, which also makes it useful in food products because it can be used to sweeten both liquids and solids. Fun fact, it burns blue when you burn it on platinum foil, and it smells like caramel. And so this also makes it desirable for food products because not only does it make foods taste sweet, it actually makes them smell sweet when they're baking at home. And erythritol is naturally present in a variety of plant products, not only the lichen in which it was originally discovered, but also in the blackstrap molasses like we talked about, and in watermelon, pears, grapes, and in mushrooms as well. When you look at the sweetness of erythritol compared to regular table sugar, it's about 60 to 70% as sweet. So not quite as sweet, but not insignificantly so. And like I mentioned, there's no aftertaste with erythritol. And so you can use it in food products without adding any additional flavor that might not be wanted. So another interesting feature of erythritol is that it is endothermic, meaning that when it's dissolved in liquid, it actually absorbs heat from the liquid. And this results in kind of an unusual cooling sensation. So if you've ever eaten a food product that's sweetened with erythritol all by itself without anything added, it causes this sensation of coolness in the mouth, which is fine, but it's weird. You can negate this by mixing it with an exothermic product like inulin fiber that causes heat release when it's dissolved. So that combination of the exothermic and the endothermic reaction causes a net neutral thermic effect of food. So oftentimes erythritol is combined with other sweeteners or other substances when it's used as a sweetener. So the majority of erythritol is absorbed in the small intestine. It's absorbed in the small intestine. So just knowing this part, you would think, well, this can't be a non-nutritive sweetener, right? If it's absorbed in the GI system, it's going to be processed, it's going to be broken down, it's going to contribute to blood sugar. But if you look at the urine, it's actually excreted unmodified. So even though it's absorbed, it's traveling through the bloodstream, doing its thing, but it's not being broken down, it's not metabolized, and it's excreted into the urine. And this is the feature that makes it both 0 calorie, 0 carb, and non-nutritive. It goes in and then it goes right back out. So this seems pretty good, right? It's not nutritive, it doesn't taste bad, you can negate that cooling effect. What do we know about erythritol in health? And what I'm going to present to you is the 2 studies that were used by the FDA and the European Union to classify erythritol as safe for use in humans. Rats. Rats were given diabetes with streptozotocin, and then they were given erythritol for 10 days. And they wanted to see what the health effects were of erythritol in these rats. And what they found were that after 10 days of erythritol, the rats had lower blood sugar, and in their organs, specifically liver and kidneys, they had lower glucose levels in those organs when the rats were evaluated. In addition, they also looked at markers of oxidative stress, and they found that these were lower in the rats who had consumed erythritol, and they also had lower serum creatinine. And so based on this information in the rats, the researchers concluded that erythritol can improve damage caused by oxidative stress and diabetes. So good stuff, right? This supported the finding that erythritol was safe. But as we know, rats are not humans, and so they wanted to look at this finding in humans as well. A pilot study performed in 24 people with type 2 diabetes looked at what would happen if they had erythritol every single day for 4 weeks. And they looked specifically at vascular function, and they used fingertip peripheral arterial tonometry to look at blood flow and vasculature. And they found that after consuming erythritol for 4 weeks, all the participants had decreased central pulse pressure and decreased carotid femoral pulse wave velocity. So it seemed like erythritol caused a decrease in blood pressure and improved vascular health in these individuals. So the study concluded that erythritol improved small vessel endothelial function and central aortic stiffness, and not only is safe, but might be the preferred sugar substitute for individuals with diabetes. So if you're looking at sweet and low, if you're looking at NutraSweet, if you're looking at sucralose, if you're looking at stevia or erythritol, maybe pick erythritol based on this study. So these are the 2 studies that the national and international organizations used to classify erythritol as safe. Seems fine. So erythritol is really good, right? But we have 2 studies, one done in rats and one done in humans, and we're scientists, and we want to know if this is actually safe for the long term, right? Both of these studies looked at a short term, 10 days and 4 weeks. So we need some more information. And the scientific community agreed with us, and they said, let's do some more. Let's look at erythritol in people without diabetes. And so they found 264 college freshmen, and they wanted to see what erythritol levels looked like and whether this could be correlated to health effects. So they did not give these college students erythritol, but they looked at their baseline levels of central adiposity, A1C, and blood isotope tracer studies, looking at erythritol in the human body to see if they could correlate those levels with health outcomes. And one interesting thing that they found looking at these isotope studies is that we as humans actually make erythritol. It's synthesized within our own bodies through the pentose phosphate pathway. And they found that these college students were making erythritol. They also found that those who were making more erythritol had increased central adiposity by about 10-fold the times the people who had lower erythritol levels. And then they also found that those who had higher erythritol levels had a 21-fold times likelihood of having a higher A1C. Please note this is an A1C greater than 5.05%, which is still normal, but this is the category they broke it down into. But the higher A1C was correlated with the higher endogenous production of erythritol in these students. So they concluded that endogenous erythritol levels may contribute to obesity in young adults. So this is a study of endogenous erythritol, but interesting and different findings than they determined when it was given to people. This is a second study that was published in 2018 looking at metabolomic profiling of just over 2,900 individuals who had been enrolled in the atherosclerosis risk and communities study. And what they did here is they wanted to look at erythritol levels and compare this to the incident development of type 2 diabetes. Is there a relationship between these two? And they found that people who had higher erythritol levels had a 1.66 hazard ratio for developing type 2 diabetes. And so they said, well, higher erythritol levels noted in the serum, not because they were given erythritol necessarily, they weren't prohibited from ingesting it, but the levels were associated with a higher risk of incident type 2 diabetes. So erythritol is bad. We should not have erythritol. It contributes to adiposity, it contributes to higher A1C, it may contribute to type 2 diabetes, but these were both association studies, right? And so we know that association does not prove causation. So we need some more information. And this is the context in which the Nature article was published. We're using a lot of erythritol. People are using it for all different purposes. And they wanted to know, is it really safe? And what are the health effects? And so the publishers of this journal or this study really wanted to take a deep dive into erythritol. And this is the article. And when this came out, everybody, except for Dr. Watson, because she did not know about this, but everyone except Dr. Watson was freaking out. So USA Today says erythritol, it's common in keto foods. It may increase your risk for stroke. CNN says erythritol is linked to heart attacks and strokes. Wellness says it's linked to heart attacks and strokes. Health says it can make your blood stickier and cause an increased risk of stroke. And then Heart Health also says the same thing. And so people were throwing away their erythritol sweetened products. Erythritol stock went down. This was a big deal. And so my brother-in-law reached out to me and he said, did you read this article? What do you think? And I said, no, I didn't read it and I don't care. And I'm going to continue to have erythritol. And then I realized that was irresponsible, both to my patients who are using it and to myself who has many products in the pantry. And so I took a look at this article and I wanted to share with you all what I found. So this is a big endeavor. This is not only one study, but it's a combination of 3 different parts of studies that the researchers did looking at erythritol levels, associations, and then they did some clinical data as well, looking at erythritol in vivo and in vitro. So we're going to look through all of these. The first part of this study was what they called their discovery cohort. And they did untargeted metabolomics, looking at a variety of different polyols in serum and their relationship to various health problems. The second part was a validation cohort where they looked specifically at erythritol as a polyol and its relation to incident development of cardiovascular disease. And then the third part of this study was a series of prospective pilot interventions where erythritol was used in various forms to look at platelet responsiveness, thrombosis potential with platelets, and plasma levels of erythritol and whether this was correlated with any sort of health outcomes and the previous platelet data. So in the untargeted metabolomics study, they looked at samples from just over 1,100 participants who were undergoing diagnostic cardiac evaluation. So not necessarily well people, not necessarily sick people, but a cohort of individuals. And they looked at these people for 3 years prospectively into the future. So 3 years of outcome data compared to 10 days and 4 weeks. So this is definitely the longest study. And what they wanted to do was determine the risk of major adverse cardiovascular events, or MACE, compared to a variety of different polyols. And what they found were that erythritol levels were associated with MACE. They specifically broke the erythritol levels down into quartiles. And so you can see on your left-hand side of the screen, quartiles 1, 2, and 3, there still was incident cardiovascular disease, but if you look at quartile 4, there was a significant increased risk for cardiovascular events in those who had the higher erythritol levels. And then on the other side, you can see the adjusted ratio or the adjusted data for cardiovascular risk factors. Quartile 4 was still statistically significant for the onset of MACE in those with higher erythritol levels. And so what they concluded with this was that erythritol levels were associated with an increased incidence of MACE, particularly in quartile 4, and they calculated the hazard ratio to be just over 3. So 3 times higher risk with the highest erythritol levels. So this doesn't look good for erythritol, but these were untargeted metabolomics. And what this specifically means is that it's semi-quantitative. And this can be a problem specifically with polyols because polyols have structural isomers that can be very similar, and an assay can detect a variety of different isomers as opposed to just a specific one that you're looking at. And here's an example of different polyols that are different isomers, and they may have different effects. And so what the study authors wanted to do was to make sure they were looking at the isomer of erythritol that's used in food products. And there was no assay to detect that. So what they did is they developed an assay. They developed a stable isotope dilution that was able to be processed through LCMS to both separate erythritol specifically, the one that's used in food products, and to quantify it. So they could be certain that this association was related to the clinically relevant erythritol molecule. And then they wanted to look at 2 different cohorts, again, with longitudinal data. So they selected 2 whole different cohorts of individuals, one from the US, just over 2,100 participants, and the second from Europe of just over 800 participants. They enrolled them at quaternary referral centers, and they looked at their erythritol levels and then documented both the baseline prevalence of cardiovascular disease and other cardiovascular risk factors, and then they looked at them prospectively over that 3-year time period to document the incidence of MACE. Here is a graphical representation of what they found, both in the US cohort, which is on your left-hand side, and then the European cohort, which is on the right. And they found that, again, in quartile 4, the highest levels of erythritol is detected with their fancy assay, there was a higher baseline prevalence of hypertension, diabetes, higher A1C, lower GFR, higher BMI, higher incidence of baseline cardiovascular disease, a higher likelihood that they had had a myocardial infarction in the past, higher LDL levels, lower HDL levels, and higher triglycerides levels at baseline. And then they found that, again, the risk for major adverse cardiovascular event was higher in the people who were in the higher quartile for erythritol levels. You can see on your left-hand side, again, quartile 4 is in the red, and that risk is statistically significantly higher than those in the other 3 quartiles. And then on the right-hand side, this again is adjusted for those other cardiovascular risk factors that we talked about: diabetes, history of MI, cholesterol levels, A1C, and after adjustment, quartile 4 still had a higher risk for incident cardiovascular events. So they said that these higher levels of erythritol were associated both with baseline prevalence of cardiovascular disease and incident major adverse cardiovascular events over that 3-year time period. And the 4th quartile, like we saw, had the highest risk compared to the lower 3 quartiles of erythritol levels, and these risk factors remain significant even after adjustment. They were able to quantify with their assay that every 1 micromole increase in erythritol level increased the risk by 16% in the European cohort, and just a little bit more, by 21% in the US cohort. So they've determined an association between erythritol levels and cardiovascular disease. But again, association doesn't prove causation. And they said, well, maybe there's something else going on that's causing this association. Maybe it has nothing to do with erythritol, but is there something different about the effects of erythritol on platelet function that could be contributing to cardiovascular disease? And so this is where they did their clinical, basic science translational trials looking at the effect of erythritol specifically on platelet function. So the first thing they asked was, does erythritol affect the function of platelets? And so they took some platelet-rich plasma from healthy volunteers and they incubated it with a physiological level of erythritol, similar to what they detected in the metabolomic studies. And then they used 2 controls, glucose and 1,5-anhydroglucitol, to compare to make sure there wasn't any confounding from the erythritol specifically. They then stimulated these samples with two substances known to increase platelet responsiveness, ADP or adenosine triphosphate and TRAP6, which is thrombin activated peptide. So they incubated the plasma samples and then they stimulated them to try to cause some effect with platelets. And this is what they found. So looking at both the ADP and then the TRAP, the two platelet adhesion activators, you can see erythritol is in the red and then the vehicle, the control, is in the purple. And for each activation, they did find that the samples that had been incubated with erythritol had higher platelet reactivity compared to the control. And this was all statistically significant. So what they determined from this is that erythritol dose independently enhanced the aggregation of platelets when the platelets were activated by prothrombotic substances. They did not see the same effect with either glucose or the 1,5-anhydroglucitol. And so then they said, well, this does seem to suggest a cause and effect relationship, at least in regards to platelet responsiveness. But we know that cardiovascular disease is not just related to platelet responsiveness. And so then they wanted to see, well, does it cause increase in platelet aggregation? Does it also cause the platelets to congeal together to potentially contribute to cardiovascular disease? And so they said, does it directly inhibit the function of the platelets? And again, they got platelet samples from healthy volunteers and they incubated them again with controls and erythritol. And again, we can see erythritol in red, the control substances in the purple. And for each substance, the thrombin and the ADP, erythritol caused an increase in platelet responsiveness and aggregation compared to the control substances. So they said, and the specific thing that they were looking at as far as platelet responsivity was the intracellular cytosolic calcium concentration. Thinking back to our physiology, we know that muscle contraction happens by an increased influx of cytocalcium. And this is how platelets work as well, as that increased intracellular calcium causes that activation. And they saw this, I'll go back one slide, looking at this on your left-hand side, the erythritol was higher for intracellular cytosolic calcium compared to the control. They also, yeah.
Now, did they make sure that the concentrations that they used in their in vivo system was similar to the concentration?
In what they detected in the, yes, these were all physiological levels. Yeah, so not a level that you would see when someone ingested erythritol. It was more similar to the endogenous pentose phosphate production like they had seen in the earlier studies. Yeah, great question. Yeah, so physiological levels of erythritol. Yeah, thank you for that. So what they also found was that dose-dependent enhancement and multiple platelet activations were seen with the erythritol like we saw in the previous slide. And this, again, was not seen with either the glucose, the control saline, or the 1,5-anhydroglucitol. So not looking good. But does erythritol contribute to clot formation? Is this a clinically significant finding, or does it just cause platelets to be a little bit more excited? So they wanted to look at that specific clot aggregation effect of erythritol. And so what they did is they took vessels from humans and mice, like we'll talk about, and they used a microfluidics device to cause shear conditions or to cause vascular damage. And they wanted to see what happened with platelets. So they took some healthy human vasculature samples and they assessed both the platelet adhesion, how sticky were the platelets, and how quickly did it take for the clot to form. And they also did this in a mouse model of carotid artery injury. It is not a good idea to cause carotid artery occlusion in humans, but it is more acceptable to do this in mice. And so they were looking specifically at how long it took for the entire carotid artery to be occluded. So they looked at the rate of clot formation and then that time for complete cessation of blood flow. And so this is a graphical representation of their findings. On our left-hand side here, you can see the amount of clot formation looking both at the erythritol and the vehicle. And you can see there's a lot more green with erythritol. That means a lot more clot formation. And then on our right-hand side, you can see the platelet adhesion measurement, and you can see erythritol in the red and the control in the purple. There was a lot more platelet adhesion in the samples that were incubated with physiologic levels of erythritol. And then when we look at the carotid artery occlusion studies, on our left-hand side is actual visual representation of that clot formation in the arteries. And so you can see at 4 minutes, 6 minutes, 8 minutes, the artery is getting fuller and fuller and fuller of clot until it's entirely occluded. And with the erythritol, which is in the red in the middle, you can see that it took just over 9 minutes for that to happen, compared to the vehicle and the 1,5 anhydroglucitol, where it took 13 or 14 minutes for that to happen. And then on the right-hand side, again, we can see that the erythritol in the red caused a significantly lower amount of time for that blood vessel to clot compared to the control or the 1,5 anhydroglucitol.
Good, I'm sorry.
No, you're good.
Yeah, now, this was added in vitro or?
This is in vitro, so these are
It wasn't with people that had eaten.
No. Yeah, these were just samples added to the artery.
Okay.
Yeah, you're
But
exactly
the
right.
second, if they did the second, that would be more impressive.
Yes, you're exactly right. So we'll talk about people in just a minute. Yeah, so this is still all in vitro studies. And so what they determined from these in vitro studies is that there was significant enhancement of collagen-dependent platelet adhesion and thrombus formation, both in the human vessels and in the mouse carotid arteries. And there was a significant reduction in the time to cessation of blood flow in the samples that were incubated with erythritol compared to control. So this is not looking good, but as you so beautifully clarified, these are in vitro studies. These are not in vivo studies. These are not looking at the actual whole mouse or the actual whole human, but they wanted to do that. And so they developed a clinical pilot study to look at erythritol levels in humans, and we'll talk about that part of the study next. So they wanted to see, based on what they determined were physiological responses to erythritol levels that they were able to detect with their specific assay, what happens after we eat erythritol levels? Or after we eat erythritol, what happens to our levels? And so they wanted to do a real-life experiment with dietary ingestion. They enrolled 8 very brave, very healthy participants, and they gave them a drink containing 30 grams of erythritol. And the reason they chose 30 grams is that using their assay, they were able to determine the typical erythritol content of various food products. And so this is a level that's comparable to an energy drink, a pint of keto ice cream, or any other typical substance that someone might consume that contains erythritol. And then they measured the post-consumption erythritol levels in the serum for 7 days after the ingestion in these participants. So this is legit. So this graph here takes a look at the baseline erythritol levels, what happens right after the ingestion, and then follows them over time for all 8 participants. And a few things to point out is that at baseline, all the levels are lowish. They immediately go up after ingestion, after about 30 minutes, and they remain elevated for at least 2 days and then begin to come down. And it takes about a week for them to reach their baseline level. So the red lines on this graph are equivalent to the various erythritol thresholds that were found to cause that platelet aggregation, activation, and clot formation in the in vitro studies that we talked about earlier. You'll notice that it's 4.5, 18, and 45. And the erythritol levels in our serum goes up to almost 10,000. So quite a bit higher when we ingest erythritol compared to those physiologic samples that were incubated. So again, erythritol levels low at baseline, increased by a lot 30 minutes after we consume it, and remain elevated for 2 days above those thresholds that were found to affect platelet function. So that's what the study shows. And what does this add? What does this tell us about erythritol? What does this add to our knowledge? And I think we can definitely say that there is an association between major adverse cardiovascular events and erythritol levels. I think they've shown that. And they've also shown that based on their specific assay, there is a higher baseline level of cardiovascular disease and a higher incidence of cardiovascular disease over time. The in vitro study showed that it did seem to be a causation between platelet aggregation, an increase in intracellular cytosolic calcium concentration, and increased platelet activation phenotypes in the samples that were incubated with erythritol. There was also enhanced collagen-dependent platelet adhesion and thrombus formation when vessel injury was caused in the samples. And there was a reduction in time to cessation of blood flow in the carotid artery mouse model. And in the human studies, when they ingested erythritol, plasma levels did increase significantly and remain elevated for at least 2 days. So this is a summary of what this study showed. So what do we do with this new information? Before this study came out, we said, it's fine. Erythritol's fine, it tastes fine, it's well tolerated, doesn't seem to be cardiogenic, has antioxidant effects, there's 0 carbs, 0 calories, and the government says it's safe, so it's fine. But maybe it's not fine, because the studies that they used to determine its safety were not very long, up to 4 weeks of safety data. So we can't really use this argument to say that it's fine. Still could be fine, right? It still could be. It's made by our bodies by the pentose phosphate pathway. It's found in naturally occurring substances. But maybe it's not fine because after we eat erythritol, the levels increase over 1,000 times what those are naturally detected physiologically and produced endogenously through the pentose phosphate pathway. So what do we do with this? Why is it so hard to make a decision? And I think one of the reasons is that with all non-nutritive sweeteners, there are not a lot of randomized controlled trials. And when we don't have clinical data, our judgments and our recommendations are based on expert opinion. There are a few meta-analyses looking at erythritol, but the results are a little bit conflicting. So this first one that I'll tell you about was published in 2017, and they looked at all the randomized controlled trials of erythritol, and they said it does not provide the intended benefits. And indeed, based on the trials that they looked at, they said that erythritol was associated with weight gain, with type 2 diabetes, and with cardiovascular events. But this meta-analysis, again, looking at the randomized controlled trials, published in 2014, said it's associated with weight loss. So erythritol can cause weight loss. Excellent. And then this third meta-analysis published in 2019 looked at all the randomized controlled trials and said there's really no difference between people who use erythritol and people who don't use erythritol. And the authors of this meta-analysis went on to say that most of the studies that they looked at really didn't have very many participants. They were a very short duration, and the methodology and the reporting quality were limited. So basically, they said there's just not enough data to really determine the effects of erythritol. So when we're looking at non-nutritive sweeteners, it really is difficult to know whether they are safe, whether they are effective, and whether we should be using them. And one of the reasons for this is that a lot of the studies looking at them use aggregate data, meaning that they use non-nutritive sweeteners all together. Sucralose, aspartame, saccharin, stevia, erythritol, all lumped together as non-nutritive sweeteners, and so they're not broken down by chemical or by substance. It's also really difficult to determine how much of a non-nutritive sweetener is in a food. How many milligrams, how many grams of NutraSweet is added to a packet, or how much is in, our sugar-free Red Bull that we're drinking, because they're reported as artificial sweeteners. So you really don't know what we're putting in there. And there are not any studies prior to this Nature article that looked at a correlation between non-nutritive sweetener intake and blood levels. We just don't have that data. Right now, the FDA also doesn't require the reporting of erythritol content in foods. It requires that we say it's in there, but it doesn't require food and drug manufacturers to say how much you're putting in. So if we don't know how much erythritol is in something, it's really difficult to correlate that with any sort of health outcomes. And like we showed before they developed this specific assay, you couldn't even really detect the clinically relevant erythritol molecule in food. We can do that now based on the assay that the authors developed, but before, you really didn't know. So what do we do? And this is my expert opinion based on all of the data that I've reviewed and the data that we've discussed together. A few caveats. In this Nature article, the metabolomics profiling was performed one time in each of the over 2,000 participants using a fasting study. A lot of the cohort participants had underlying cardiovascular disease, and as we saw in the targeted metabolomic studies, they had a lot of risk factors. So this association could potentially induce some confounding. Do people who have baseline health concerns, particularly cardiovascular risk factors like obesity, like prediabetes or diabetes, like elevated cholesterol, like obesity, do they use more erythritol because they're trying to decrease their risk for future events? Do they use more erythritol because they're trying to follow a low carb or a low sugar diet? And so is this more of an association versus a causation? And I don't think we know the answer to that. We also know that cardiovascular disease is multifactorial. You don't eat a hamburger and have a heart attack because you have done that. You don't consume keto ice cream and have a stroke. It's not a cause and effect. There are a lot of factors that contribute to cardiovascular disease. So it's really difficult to tease out whether, and if so, how much erythritol may or may not be contributing. So what do? What did I tell my brother-in-law? What do I tell my patients? What did I do with my erythritol products in my pantry? And I think we just have to have the conversation, right? So what this journal article shows is that there may be, based on the platelet aggregation studies, an increased thrombotic risk after someone ingests erythritol. But we also need more data. We need to know if this is clinically significant, right? And I think part of what we can do to move in that direction is to establish reporting requirements for non-nutritive sweeteners. How much of these are manufacturers putting into our food products? And that can help us to determine what are the typical daily intakes? How much are we actually consuming on a daily basis? And what are we consuming? That can help us to correlate the amounts that we're consuming with our circulating blood levels. If we drink a sugar-free Red Bull, I don't know why I'm picking on Red Bull, but if we do such a thing, what level of erythritol or what level of non-nutritive sweetener can we expect to see in our blood? And once we're able to do that, then we can really evaluate the clinical effects of circulating levels of superphysiologic substances in our bodies. And then we can look at safety studies, looking at the long-term effects, because people eat food every single day of their life. It's not a 4-week time period. It's not a 9-month time period. It's not even 3 years. We're doing this for our entire lives. So looking at those long-term clinical outcomes is something that is going to be important if we really are going to give recommendations for this. So, do we have to choose between cake or death? Can we have our cake and eat it too? Should we sweeten our cake with sugar or erythritol or something else? And so I am going to move this video clip forward so we can talk a little bit more about this. So most of us are going to choose cake if the option is cake or death, except for those of us who choose the chicken. But I really do think that with every clinical question that we have with our patients, really being aware of what data is out there, what are the limitations, and what can we tell them about health risks of anything that they ask about, whether it's non-nutritive sweeteners, whether it's nutrition, whether it's medications. And I think we need to be really honest with our patients and with ourselves that sometimes we don't have the answers. We know that cake sweetened with sugar and processed foods in general are associated with clinical health outcomes that are damaging to our patients. And so I think telling our patients that limiting processed foods, limiting added sugars, limiting simple sugars is the right thing to do. But I do not know that we can advise them either positively or negatively about erythritol. I will tell you that I still have erythritol containing products in my pantry and I still use them and I still eat them. But I do think about this study and I do recognize the fact that I am doing it taking unknown risks and that more data is needed. So with that, thank you so very much for joining me today. And I would love to take any questions.
So in the study that you had with the human study where the levels went up, you said that they were given what people would normally consume. So it went up. So if I'm consuming it every day, then my level will be above 5,000 all the time.
Yeah.
Correct?
Yep.
You know? So none of the studies looked at that level all the time. It might not do the same thing. It may just be something that happens.
Yeah.
When you go from 0 to this, and nothing might happen.
Yeah. Yeah, you're exactly right. You're exactly right. So there aren't any studies looking at erythritol ingestion over the long term. This is the first
that
one.
Study, did they look at platelets at the end of the 1 day or 2 days?
Did they? I'm sorry. Did they look at what?
The platelet aggregation.
No. The platelet aggregation studies, those were just in vitro looking at incubation with erythritol. They didn't look at the levels because they gave the level. So that was totally separate from the ingestion study.
Yeah, I would say, what we know for sure, and the original workers were like Russell Ross and Joe Raven. People don't talk about them anymore, but that was the work was done 20 years ago. But anyhow, what we know, I think, is that excess fat is toxic. I consider it to be toxic. Okay, just like a poison, just like smoking cigarette, because we know with these atherosclerosis, we know it leads to a metabolic syndrome and all those other things. It can cause diabetes in many people. And so I think you really have, in my mind, I focus on excess fat in body weight, BMI, which they say isn't the best index, but it works for most people. And so, I think unless you have some really good evidence of something substantial, and again, they're not comparing this with what would happen if people were eating an equivalent amount of sugar. You see?
Yeah.
So I think you have to kind of take it in that vein. And what I tell people, and they pay no attention to what I say, of course, is that you've got to keep your body weight down. You've got to watch your calories, and that's the most important thing that a person. So I've read a number of these things by Hazen, and I don't know that I paid as much attention to it as you did, but it just seems to me if you swap an artificial sweeteners for sugar, you're better off.
Yeah, I think limiting our intake of processed foods in general is probably the best thing to do, and breaking it down by different substances. I think we just don't know, but I agree. Making your nutrition plan as healthy as possible is always the right thing to do. Dr. Watson, you had a question.
Well, more of a comment, but I think this is fascinating for a couple of reasons. primarily because of the impact on the platelet aggregation. So in my mind, I add this to the long list of plants that we know directly interact with the coagulation cascade. And so I wish they had done a simple plotting time on as many blood samples they took in the individuals in the pilot study at the end, because I think it would have been fascinating to at least get some indication on if there's a signal that this is being affected in vivo as well. So I think that kind of jumped into the yet another plant that causes problems.
Yeah, yeah, I think there's, yeah, just so much more work that needs to be done before we can make a definitive.
So is it
Call.
possible the mechanism, it's not metabolized, is it bind to something or is it protect?
Oh, they said nothing about the
There's something that's just
mechanism.
Going through your body.
Like it's going through unaltered. Why is it caught? Yeah, yeah.
It's sticky, is it changing charge in this particle?
Yeah, the mechanism, the only mechanistic thing they determined was that there is an increase in the intracytosolic calcium, but they did not propose why that occurs.
So I want to know what is in your pantry that has got this.
Oh, she showed it.
I have, oh
I
man.
missed the
So,
first part.
oh yeah, so I've got, I have like a white sugar substitute, a brown sugar substitute. Dr. Thrasher, my protein bar that I had for lunch did have erythritol in it. I shared some gummy bears during a recent conference that did not have erythritol in them, but quite a few kind of dessert sort of products in my pantry. I have other things also, but yeah, there definitely is. There's some risk happening in the Folsom household.
So there any questions on that?
Let me check. You had one more comment. Sorry.
Yeah, I would just point out now, that in the graphs you showed, they showed 3 quartiles that were almost right next to each other, and then they showed a 4th quartile, which showed a difference. Okay?
Yes.
And then they made a linear relationship to say that if you ate so much, you were increasing by so much. But of course, that's not true, because all of that whole thing was affected by the 4th quartile, not by the first 3.
Yeah, and so these were not ingestion. These were just
Well, yeah,
baseline
even no
fasting
matter how it was
levels.
done, there was no linear relationship.
Right. Yeah.
Is what I'm saying.
Yeah, these are just one-time.
And because that's a
cross-sectional.
statistical technique that people do all the time, where the 4th quartile is the one that's very high, and all the others are very close to each other. But then they say, well, see, there's a linear relationship. But there isn't.
Yeah, it's just association studies. Yeah. So we have some things in the chat here.
Yeah, a lot of
transcript. Yeah, no, there's a lot of-
It records everything.
Oh, this is okay. Hold on. Let me
Yeah,
go to current. Oh goodness. Okay, there's a lot of.
Go to the chat.
Oh boy. Okay. Oh yes, here's the chat. Okay, this is what we want. Okay. Oh, so someone couldn't see and couldn't hear. Oh, but oh no, that's from last week. Let's see. Okay, so someone couldn't see, try to restart, can't hear. Okay, I see no questions in the chat. Does anyone online have any questions they would like to put into the chat? We'll give you-
Lisal, this is Swan. Can you hear me?
Oh, I can. Dr. Deo, I hear you.
Hi, wonderful talk, hot and exciting topic. Lots of good information, so thank you very much. So, just comments and just some opinions, I guess. So one thing is, I think there is a need for sweet something in everybody's diet, which basically to the development of all of these or introduction of all of these non-nutritive sweeteners, I think. So my first thing is, I really think that as a country overall, the food habits, if they could be different in the sense that not needing to have a dessert with every meal, that would help mitigate some of these problems, because if you had non-processed foods and in moderation, there would really not be a need for a non-nutritive sweetener, and you could just use your natural foods example, if you want to just get dates or something in your diet that could add the sweetness, if you don't want the regular sugar, that would be one way to handle this. But the fact that almost everybody wants to have a dessert with every meal, I guess, leads to the need for these non-nutritive sweeteners. I think even with the kind of not so concrete evidence that we have, it looks like the artificial sweeteners do have side effects, and I guess it's just a matter of time before we have more evidence to more problems with these. So why not just tell the patients to use a regular either sugar or sugary foods that can add to the sweetness and not have to worry about this?
Yeah, I think that's a really good point. So Dr. Deo was saying how we in our society think that we need to have dessert at every meal. And so we use a lot of artificial or non-nutritive sweeteners so that we can do that without adding the extra carbs and the added sugar. But what if we didn't have dessert at every meal? Or what if we had things that are naturally sweet, things like fruit or things like dates, and that would limit our need for using non-nutritive sweeteners in general. And I think, again, moving towards a healthy nutrition plan is a great idea. Very good. Anybody else with any other questions? I think that's it. All right, everyone, thank you so much for coming today.
That was a wonderful talk.